What are the possible empirical paradigms that can investigate the relationship between the structure of qualia and the information? So the relationship between structures of qualia and the information needs to be investigated in paradigms that can manipulate one or both while measuring one or both, all right, qualia and the information. And first question that I briefly want to go through is that why is our visual experience of color, motion, phase are so different from each other? And most likely that is likely to be related to how these neurons within V4, V8 for color, MTV5 for motion, and phase, FFA for phase are different. That's the first conjecture. So what do we know about the color qualia similarity structure? So that already gives us some kind of hint of what we should expect for the color, qualia generating kind of structure like V4, V8. So for example, no two color qualia in the same space-time is experienced, right? You usually cannot experience, or even with the illusory kind of situation, on the same spot at the same time, you can't experience red and green at the same time. There is a situation where it's amorphic, like binocular rivalry, it changes over the time. But at the given time, to experience both is not that, easy to at least imagine, and also possibly impossible to induce. And this is probably the similar kind of situation like taste, smell, and touch, all of these things, when you trying to, mix two things, then you can't just separately experience two things at the same time. And then this integration happens so strongly that, the two component things starts to get experienced as another new qualia. And this is quite different from other things like visual motion, the face, or sound. The visual motion, for example, at the one particular location, you can have both left and the right, top, and then you can see a bunch of the dots moving into top and then left, at the same time. But there are some interesting kind of situation where you can't perceive both up and bottom at the same time. That's a very interesting kind of phenomenon observation. Same goes for face and sound. Usually, all of these kind of things are possible to experience at the same time. Face, if you transparently superimpose two face, you can experience both at the same time. Sound, same. C and D can be experienced at the same time. And then another thing is that this color qualia is lost. When it's lost locally in space. Like, in some of the patients who report the loss of the color qualia, they lose the color sensation in particular visual field, like here, top right, all of them, all the color gets lost. And it's not like, loss of only red or only green. And it's probably related to this relational structure of the color. The most likely the neurons that has to generate a color qualia are structured in a way that one of them cannot be only lost, but it can be lost in a situation like in the case of concepts or the other types of the qualia, such as face. Face can be perceived as eyeless, like here. You can experience it as such, but that's not the case, for the color qualia, right? So this type of the property of the each of the qualia has some kind of implication for neural circuitry and the causal structure. And in the case of face, in the Mona Lisa, you have this distinct kind of compositional structure, relationship, eyes and mouth and nose are all each perceived as it is, but also composition is also giving some specific kind of experience in its own. But at the same time, we experience them as whole, and these are probably related to the way that is that FFA lesion or somewhere that is responsible for face qualia, most likely right FFA, is configured. And then that possibly, if IIT is correct, then by looking at the information structure of FFA, maybe we can find the correspondence between the composition of the phase qualia and the composition of the small integrated information in FFA. And then another potential paradigm to address this type of question is, why is our visual experience so different from our auditory experience? And it sounds like a really vague question, but and so you might ask, can we address this empirically? But I would say it's confidently yes. And using a rewiring paradigm or learning paradigm, we can probably address this empirically. So what is a rewiring paradigm? The rewiring has been developed by the biologists, neuroscientists, where they change the way the connections are made within the brain through the birth developmentally. So Mrigan Ksar at MIT published a series of the paper in Nature to demonstrate this. As you learned in this course, the normal brain, the mammalian brain, has an axons starting from retinal ganglion cells and then arrives at the LGN thalamus and also projects to speria colliculus, right? And then the speria colliculus pathway goes to pulvinar and then also the cortex to guide the visual blindsided behavior, if you remember. And then LGN pathway projects to visual cortex, and then that is supposed to generate a visual phenomenology. And if you distract visual cortex, then you become blindsided. You lose the phenomenology. And then we didn't cover much about the audition, but audition, we also know that the ear input, sound input to the ear goes from inferior colliculus and also another pathway to go through MGN. That is another pathway, another part of the thalamus, like LGN, medial geniculate nucleus. LGN is a lateral geniculate nucleus, and then that goes to auditory cortex. Okay, that's the auditory pathway, and that usually doesn't interact at all between the visual and the auditory. But using ablation technique plus some kind of chemical technique, what is known, at least in the case of ferrets, the small animal, what they can do is to induce the connection from eye to arrive both LGN but also MGN. And then you need to cut this speria colliculus actually. And then we can also cut the connection from ear to inferior colliculus and also MGN. And then eventually that establishes this new connection from MGN to visually or responsible auditory cortex. So this is in addition to the normal visual cortex through the LGN. Okay, so that is called a rewiring paradigm. And that has been done in the animals, but also, to some extent in human, sensory substitution paradigm has been implicated to be related to something like this. But we don't know exactly how the sensory substitution in human works. And I'm going to talk a little bit about it later. So the critical experiment that Mrigan Ksar's group has done is the 4-phase experiment. So it's quite complicated, so I'll go through one by one. Okay, in the phase 0, these rewired animals that became adult, the surgery and so on was done in the at the birth, and then the ferret becomes adults. Okay, then their left visual field receives the input through the LGN and speria colliculus, and then projects to visual cortex, and that's a normal control cortex. Okay. And then also, auditory pathway, is very, non-lateralized. Any kind of stimulus go from right side or center or left or whatever gets processed by, our ear that locates here on both sides, and then also, both of the cortex processes it. So there's no point in talking about, left and right. So when the sound comes from this speaker, then the ferrets were trained to go to this auditory reward spout. So they put their, head here and then wait for the, stimulus. And then when the sound comes, beep, then they go to, auditory reward location. That's the training phase, phase 0. Okay, and then also another situation of the phase 0 training situation is that when the light is flickered presented on the left side, okay, then the animal ferret needs to go to this visual reward spout. And again, just to make sure that the left visual field stimulus is always only stimulated through the visual cortex. And here is the boundary of this binocular field that you also can have this binocular field. Both left eye and the right eye can see central part of the vision, but there are some peripheral part that your left eye can only see, but not your right eye. And that's the location of this left-right location. Left-right thing is happening. But here, the ferret can learn this left-right to the visual spout and sound to the left auditory spout. That's really easy. Vision versus audition. It's so easy to discriminate. So animal doesn't have any problem. So the response is almost 90% to go to the visual spout, right? And just to make sure, the last one, the sound wise, they go to the sound part, almost like 90% accuracy as well. So that's a phase 0, where they were trained to learn the rule of the audition versus vision, but vision using the one that used visual cortex only. Okay, so now in the critical phase 1, okay, for the first time, they are tested with sound that happens at the center and also right. And then the critical thing is a right. So, I'm going to talk only here. Right visual field input goes to LGN and MGN. And presumably that evokes a response in auditory cortex and visual cortex. And in this case, there's no wonder, but ferret reports, oh, I saw the light here. So the percent visual response becomes almost like 100%. Here, it's kind of confusing graph, but you don't need to pay attention to this left side. The left side axis is only for the left part of the graph. And this is a pre-LGN LP region. So here, when this guy, LGN LP pathway is intact, both visual and also auditory cortex are activated. And so it's not super, interesting on its own, okay? But this is the important thing is that ferrets were never trained or rewarded to say that it was a vision at this point, but they spontaneously reported always, the vision. Second, critically, when they removed the LGN LP pathway in the phase 2 experiment, so here we no longer have this visual cortex anymore because they lesioned it. So the right light has been transferred into MGN and then processed in the auditory cortex. And only with the auditory cortex, still they reported this post LGN LP lesion, right light. They still reported that seeing vision. That's a really critical phenomenon, right? So auditory cortex appear to seem to generate this response. But if you are really careful, you might still say that auditory cortex may be just making some kind of association with the visual cortex through this always parallel input and so correlation and things like that. Then to address that, what they did was the final critical experiment where they now remove this auditory cortex. And the visual cortex is still intact. So when they did that, then the response to go to the visual or auditory becomes completely random, 50%. So that means that the post A1 lesions, they don't now see and report vision anymore. And that's the 4-phase experiment. Phase 1, 2, 3 is the critical ones, okay? So just to summarize this rewiring experiment, which is a bit complicated. First, in the training phase, the ferrets learn to respond to associate vision with the right spout and the audition with the left response spout using the control hemisphere that is intact, okay? And then in the first phase, before the LGN-LP lesion, they reported seeing when they processed the light for the first time. And then in the phase 2, critically, after LGN-LP lesion, now all the light coming from a right side is only processed within the auditory cortex, and then they continued to report seeing. So then that means that light within the light response was produced within the auditory cortex, and that is supported by the third experiment where they stopped reporting seeing after A1 lesion. And then combined with these, it's quite compelling evidence that ferrets probably experience visual qualia, something visual generated by the auditory cortex. And the critical thing about this kind of animal experiments is that they can examine what's happening in the brain, right? So if the IAT or any kind of structural theory is correct, then the visual qualia has to be generated by some kind of structural rule that is governing this visual cortex. And in fact, what they did was looking at this type of connectivity. So in the normal visual cortex, I didn't explain quite in detail, but there is this orientation column property has been observed across many animals. Okay, so each of the cortical location contains similar neurons that respond to one particular orientation of the line, but not the other. And this color corresponds to the dominant, orientation that evokes a response in each of the neurons. So here, normal V1 shows this orientation columnar map, which is very, typical for many, many neurons. And then there's this, pinwheel-like situation here, where, at some special point, many of the orientation becomes mixed. And that's a sort of the topological feature of the cortex in mammalian brain. And then in each of the neuron, if you look at this, the neurons, let's say, cell bodies here, then in the case of the visual cortex, neurons tend to have a connection with the neurons, which has similar kind of a preference in terms of orientation or position or spatial frequency and so on. So for example, when you find the neurons in this location, right, then they tend to have a connection to the neurons in this location with the same preference in the orientation and here, and then here, and then here, when the neurons are located here. And that's observed in this two-dimensional map of this axonal connections of the B1. So that's critical for the visual cortical property. On the other hand, normal auditory cortical neurons have a very different types of the connection. The neurons in the auditory cortex, for some reason that we don't fully understand, they tend to have a connection to the neighboring neurons in a more or less symmetric and also very similar kind of ways. Very different from visual cortex, and that may be underlying how how we experience a space in the vision, but not in the auditory cortex, but more to do with the time. That's a sort of a conjecture. And then rewired A1. Now they start to show this, similar orientation column like visual cortex, right? But you also notice that something really weird happens. Like here, this is a bit, disorganized. This is also disorganized and so on. And that's probably because there is some kind of genetic instruction for this auditory neuron to become like an auditory neuron like this. But input pattern is more like a visual input. So that's why Rewired A1 starts to find this kind of visually invoked kind of pattern. And then critically, the auditory cortical neurons now start to exhibit the property of the V1-like, connectivity. So starting from this cell body, there is a separate sort of, connectivity of the neurons from here to here and then here and so on. Similar orientation or similar spatial frequency or position starts to get connected, similarly. And the conjecture is that this type of the different types of the local connectivity pattern supports visual qualia on one hand and auditory qualia on the other hand. So in summary, structures of qualia and the information can be manipulated independently and or jointly. Advanced molecular or neuroscientific tools enable such dream experiments, and that can be done in the future. And there are many more existing paradigms rather than rewiring. I didn't actually mention that, but some of the promising techniques include synesthesia in the human or congenitally blind people, human as well. And these people can be actually now studied with a super high resolution fMRI, sometimes 70 Tesla, or even higher. And also in terms of the stimulation, we talked about the TMS, transcranial magnetic stimulation, which excites actually many, many neurons at the same time. But ultrasonic stimulation, ultrasonic sound stimulation can target very small portion of the brain and then inhibit the responses. So that might be much better way to manipulate the brain and then see how that changes the structure of the qualia. And also optogenetics can be combined for the animal experiments, and then rewiring and many things can be done potentially instantaneously. So even in the case of the ferrets experiment, that was all developmental and so on. But potentially future experiments can simultaneously and in controlled manner for the auditory cortical neurons to become like visual neurons in terms of connection. Just with one click, and then go back to auditory cortex with another click. And if that changes the way the animal reports with combined with a similarity kind of reports, that's going to be highly convincing that these structure property of the circuit generates the structure of the information in particular way, and that corresponds to structure of consciousness of qualia. So that's a sort of the plan or the prediction or hope for the next 10 to 15 years.